The collaborative effort really enables us to take ideas and rapidly act on them by engaging all members of the consortium to contribute their expertise. And so we're able to do experiments much more quickly by distributing the effort, and also we don't have to reinvent the wheel in each laboratory and have each laboratory become expert in all the methodology that's required. And I also think it speeds the effort in that we really share our best ideas with each other and critique these ideas so that we can more rapidly hone in on the ideas that are likely to be most productive and discard the ones that are going to be less productive over time. And this is a wonderful opportunity to kind of work with other engaged people who are thinking about the same problem as you, because the back and forth intellectual component of this is, I think, really moved this forward more quickly because we are sharing things that we haven't shared with other scientists. And this, I think, speeds the development of more innovative approaches to tackling the disease. So I actually think there are two interesting things that are unique about this consortium. One is that the goal was to bring together scientists who were not currently working in the field of glaucoma, to bring a fresh perspective to really try to think about and tackle this disease, and really encourage collaboration between multiple laboratories, which was very distinct from the way traditional biomedical research is supported and funded. The other thing that was unique about the consortium was that there was a commitment on the part of the Glaucoma Research Foundation to really invest in a group of scientists and to provide sufficient support and also sufficient time to really develop the methodology and the ideas and move things forward. It always takes a little bit of time when new people enter a field to get up to speed and become familiar and really also start to apply some of those new ideas. And the Glaucoma Research Foundation was there at every step of the way and providing continuous funding, encouragement, and support for the work. And that's really made a tremendous difference in allowing us to pursue riskier ideas and take chances that we wouldn't necessarily have taken if we had been forced to pursue traditional methods of funding for this work. We need to be able to intervene at these early steps in the disease before there's significant, profound vision loss, and we need to understand the basic biology of what's happening. What are the mechanisms that are driving things forward and leading to the loss of vision? And we are focusing both on changes that are happening within the retinal ganglion cells, which are the cells that are primarily affected in glaucoma. These are the cells that ultimately die in the disease. But we're also presenting ideas that show that there's actually very interesting interactions between the ganglion cells, the retinal ganglion cells, and other cell populations in the retina, and that this is a dynamic process that's changing over time and then spreading throughout the retina as the disease progresses. And so we hope that by focusing on these early fundamental events and really getting to the heart of how these cells are interacting, we'll be able to develop therapies that may slow or cure this disease.